research, in case any parents or high schoolers are interested in getting into high school level research, and what are the lessons that I learned about doing research at the high school level. So my talk is kind of for anyone who's interested in my project, or the major topic of it, which is collective behavior, as well as anyone interested in getting advice on doing research at this level. So to kind of outline my presentation, I'm going to start with kind of giving the story of how I got into high school research. That's going to be, it's about 10 or 15 minutes. Then I'm going to spend about 20 to 25 minutes talking about my actual project and getting into the details there. Then 5 to 10 minutes wrapping up, and then if there are any questions at the end, there'll be time left for that. Okay, so what is the Science Talent Search? So it's this competition run by the Society for Science in the Public, and according to them, it's the nation's oldest and most prestigious science research competition for high schoolers. It was first sponsored by Westinghouse, then by Intel, and then I was a member of the first class sponsored by Regeneron. So basically, what it entails is you do your own research project, and then you write up a paper for it. And I should clarify, it's for seniors, high school seniors only. But you write up your research paper, then submit it, and based on your paper, that gets cut down to 300 semifinalists from about 1,700. I think it was actually a little bit more than 1,700 applicants this past year. And then from those 300 semifinalists, there are 40 finalists who get picked out. And I happen to be one of those finalists. So then what do the finalists get to do? They get to spend a week in DC where there's further judging. And that judging takes the form of one-on-one, or not quite one-on-one, but you get sat down in a room with just a few judges, and they ask you questions from all over science, and see sort of your problem-solving ability, as well as poster presentations, which are open to the public. And so those are the two portions of the judging. And in addition to all the judging and cutting it down to the 10 top winners, you get to meet all the other finalists who are incredible people and participate in fun activities like we went to see all the monuments around DC and spoke to congressmen, that sort of thing. And of course, there's cash prizes. I think the top prize this year was $250,000. Yeah, I didn't happen to be one of the top winners, but I was one of the 40 finalists who got to go to DC. So I'm somewhere in these pictures. So how did I get there? Paint, which was the project that I submitted to the Science Talent Search, was definitely not my first project. I've been interested in science for a long time, and because of that, I've been doing my own sort of little projects and explorations on the side, independent of school or stuff like that. So I think my first major project was my original programming language that I came up with the summer after 9th grade, Minima, which it was a stack-based functional language with postfix syntax. It was designed to be easy to implement while still having high-level features. But none of that, is really important right now. But the point is that I think to do research in high school, you have to be self-guided to some extent. You can, if a lot of people look for the help of mentors and research classes, and that's all definitely good. But you have to have enough passion and interest to want to put in a lot of time and effort, because it is a very it is a large time and effort commitment. So after Minima, my next sort of major project was over the course of my 10th grade year, studying tardigrades, which are these microscopic organisms that are very good at surviving extreme conditions. So for instance, they've been put into space. They've they've tested all kinds of stresses like extreme temperatures, both high and low, that sort of thing. And so the way they survive through those conditions is that they go into the state of cryptobiosis, where they shrivel up, and their metabolism gets slowed down incredibly. So they're barely alive until the stress is removed, and they receive water and oxygen again. They sort of come back to life. So I was interested in the process of cryptobiosis, and in particular, I wanted to see if they used a certain kind of DNA repair mechanism. But the problem was I could never get this project off the ground because the cultures just wouldn't survive. I just couldn't get this project started. We purchased cultures at first, and those just died out. And a lot of that, I think, was due to the climate around here. And then after we tried that for a while and just realized it wasn't going to take, we tried isolating tardigrades from the environment, but that didn't work out either. So eventually I left that project, but I think that was important because it taught me the importance of failure, because you can learn a lot of lessons from it. And you have to understand that in research, not every project is going to work out. And knowing when to say, okay, maybe I should go a different direction, maybe I should explore something else, I think is an important skill. And it also taught me the importance of mentorship, because during this whole time, I was talking a lot with my biology teacher, who was very important in getting me to think more like a scientist. It was just a really good experience to be able to talk to him and throw ideas around and try to troubleshoot what was going on, even if it didn't ultimately work out. And in fact, so I mentioned pivoting to new ideas if necessary. And what I pivoted to next, I think it happened to be a very good decision, because it was the project that eventually turned into Paint, which was the thing that I submitted to the Science Talent Search. So besides that, summer programs were very important to me. I attended Summer Ventures in Math and Science and Governor School, which are both North Carolina programs. And some of the reasons that are important to me, besides education, so we took a lot of different classes. For instance, at Governor School, I took a class on biodiversity. At Summer Ventures, I took a class on geographic information systems. And besides that, and getting to speak to these professors, I think these kinds of experiences are important because they give you the experience of living independently and seeking your own projects, as well as being surrounded by like-minded peers, which I think is incredibly important. You learn a lot from knowing bright people. So the summer after 10th grade, after I moved on from my tardigrade project, I started working on the project that would eventually become Paint at Summer Ventures. And so this is one of the models that I came up with in that time. You have a network of little nodes that are connected to each other by different color-coded kinds of relationships, and they're interacting to produce some kind of behavior. So what I learned, besides this cool modeling software and how to do this kind of thing in the first place, I had some struggles with my mentor there, my assigned research mentor. I just couldn't really communicate to him what my project was about and why it mattered. So I think that taught me a lot about the importance of communication, which is another important principle, I think, because if you can do the science, it doesn't matter if you can do the science if you can't communicate to other people why it matters, what it is in the first place, and so on. Okay, so developing Paint further. That took a lot of time and dedication. I spent most of my lunches in my junior year in 11th grade in my science teacher's room, usually alone, just working on whiteboards or reading textbooks, that kind of thing. So it is definitely a large time commitment. And then my senior year, which was the year that I wrote up the paper and sent it off to the Science Talent Search, I'd never written that kind of technical paper before. So that was quite a daunting process. And it took a lot of time and guidance. And I actually almost gave up because I didn't think that I stood a chance of really of even getting semifinalist. But I looked at the list on the day that it came out, and I'd been announced a semifinalist, and I was satisfied with that. I almost didn't pick up the phone on the day that they announced that I was a finalist. But then after that, I think that all that hard work, essentially 2 years working on this project, paid off. I found this incredible group of people who are all like-minded, just really brilliant, and they have all their own independent projects that they pursued, some with mentors, some without. And I think that was a really incredible experience. Like I said before, being surrounded by like-minded peers is something that you can learn a lot from. And I definitely learned a lot from these people. Okay. So now to get into the details of my project, which was called Protein Association Interaction Networks, PAINT for short. So I had a kind of twofold purpose. The first was to develop a model of computation within cells, which I could then apply to more general computational problems. But what do I mean by computation within biological cells? Essentially, I'm asking the two questions. First, how does the cell make decisions to adapt to the environmental conditions surrounding it, as well as can that be adapted to solving other computational problems more generally? And then my second major purpose was to argue that the cell is an example of something called collective behavior, which I'm going to get into a little later, and that a model like the one that I described above could be used to answer questions about collective behavior generally, particularly whether certain patterns tend to evolve in nature in collective behavior like whenever individuals in a large population interact together to create behavior, and what are the optimal ways of interacting to suit various natural constraints. So to kind of get into that computational view of the cell that I was talking about, the computation within the cell, I thought I'd start with this quote from Steven Pinker, which I think captures the essence of what I'm trying to get at. The typical imperative from biology is not thou shalt, but if, then, else, which of course is the typical, it's a very important idea in computer science. So for an example of how the cell makes computational decisions, I thought I'd look at the expression of the lac operon, which is a very well-studied model in biology. So what is the lac operon? It's this unit in the DNA of E. coli and other bacteria, which produces the enzymes that break down lactose. So what is the problem that the cell has to solve? Well, it has to make sure that the production of that lactose metabolizing enzyme can't exceed what it actually needs at a given moment, because that would be wasteful. So it's essentially a resource management problem. So if you kind of look at this in a table view, when there's no lactose, we don't want to express the lac operon, because then we would be producing the enzyme and it would have nothing to actually metabolize. That'd be a waste. And then when lactose is present, then we express the lac operon, which can then break down the lactose that is present. Well, we add in another factor to kind of complicate things. We add in glucose, and the cell prefers to metabolize glucose over lactose because it can get more energy that way, essentially. So what happens now? Well, when lactose is not present and glucose is not present, then we don't express the lac operon because there's no reason to. When lactose is present, when lactose is not present and glucose is present, we again don't express the lac operon because the enzymes that it produces break down lactose. They can't break down glucose, so there's no reason to here. And here, as before, when lactose is present and glucose is not present, we express the lac operon to a high degree, just as it was before. But then when lactose is present and glucose is present, we express the lac operon at this intermediate level because we'd rather be diverting our resources to expressing the enzymes that break down glucose instead. So if we kind of look at this in a table and a flowchart view, this kind of resembles kind of a logical table from Boolean algebra or electronics or that sort of thing. We kind of look at it as a binary yes or no for glucose and lactose. So we can kind of make a sort of truth table. The only difference is that for the lac operon, it's actually ternary, not binary, because there's the high, low, and none states. But anyway, so basically the cell has to answer this series of questions. First, is the lactose present? If not, then we're not going to express it because there's no reason to. If it is present, then we further ask, is glucose present? If yes, then we express the lac operon to a low degree. If not, then we express it to a high degree. And so there's this kind of feedback effect that happens, this negative feedback loop, because when we express the enzyme that breaks down the lactose, we're gonna reduce the amount of lactose actually present, and eventually it goes down to zero, which acts as a shut-off mechanism. So we stop expressing the lac operon, so it shuts itself off by decreasing the amount of lactose present. Okay, so to kind of carry this computational view of the cell further, how does the cell respond to changes in the environment? Well, typically, it happens on the basis of chemical signals. So this is a diagram of the process known as signal transduction. So we have some kind of signal here being received by a receptor, which then triggers this process, the phosphorylation cascade, which is basically amplifying the signal and as well as changing its form. And then that's, in this case, it's inducing some kind of response that changes the degree of DNA expression. So the signal transduction process can cause changes in the gene expression. So that changes the degree that various molecules are being produced, as well as the spatial distribution of various biomolecules within the cell. So what's happening here, if we look at it at a higher level, basically is that the cell is changing its internal state to reflect changes in the environment, that's how its behavior is generated. So with that idea in mind, we can treat the biomolecules, which are usually enzymes and other related factors, we can treat these as the basis of the cellular computation that's giving rise to the behavior. Okay, now we want to kind of look at it from the other perspective, another related perspective, which is collective behavior. Collective behavior is a relatively new field, and it studies systems like ant hives, bacterial community. The thing that kind of unifies all these systems is that they're based on local interactions between a lot of small individuals acting within this group, but that gives rise to behavior at the scale of the whole population. So, for instance, in ants, there's a species of harvester ant that, even though no individual ant, so when they go out to forage for food, they want to make the number of ants that they send out, the rate that they're sending out ants, proportional to the availability of food. Even though no individual ant knows the layout of the area around the anthill no ant knows where the food is or how much there is in total. Just based on the one-to-one ant-to-ant interactions, the anthill, if you look at the rate that the ants come out, is actually very good at modulating based on the amount of food in the surrounding environment. So that's one example of collective behavior. So there's some stipulations. For instance, collective behavior, like I said before, it's based on local interactions. There's no hierarchy of control. There's no like ant manager sitting in the middle who's telling each ant where to go. They just kind of act on their own, and then based on simple interactions between the agents, they communicate enough information to where the behavior at the whole scale is intelligent in a sense. It's responding to changes in the environment. So what I'm arguing is that the cell is an example of collective behavior, and the sort of intelligent behavior of the cell, its response to changes in the environment, emerges from collective behavior of the enzymes and other biomolecules that are making up the cell. So if we can try to kind of fit that into this definition of collective behavior, and interact with each other, and they're not controlled by any kind of central system. It's just one-on-one interactions, and enzyme binds to another enzyme or some other kind of protein. And their enzymes are very specific, so they only bind to a specific subset of biomolecules. For instance, the enzymes that metabolize lactose bind to lactose, but not to glucose, like I was saying earlier. So that's an example of agents interacting based on simple rules. And when you add all that up, that adds up to, to behavior at the scale of the cell. So how did I model that? How did I come up with a kind of mathematical representation of that, or at least a simplified version of that for my model? Well, I started by considering a simplified model of enzymes where they either have an active state or an inactive state, which is a fairly common thing in biology. Depending on their physical conformation, to work on other molecules, or they can't. So I have this basic model of an enzyme, and then I say that enzymes belong to a population, which is described by two properties: the active fraction, which is the proportion of the population that is in the active state, and the actual population size. So here we have an enzyme population K. The total population, this total size of the population is this N sub K. And then the number of them that are active is A sub K times N sub K. And then obviously for the number that are inactive, it's just 1 minus the active fraction times the population size. And so once we have these enzyme populations, we need to model the way that they bind to each other and induce changes. So we're going to look at these protein binding relationships. So I defined 5 major relationships that protein populations can have with each other in biology. The first 2 here are activation and inactivation, which just converts, activation converts inactive enzymes to the active form, and then vice versa for the inactivation. So that's going to affect the active fraction, A of whatever the target population is. And then we have degradation, which decreases the population size. You can imagine, say for instance, one enzyme is breaking down another, it's metabolizing it somehow. So that decreases the population size. And then, so kind of you might ask, what's opposite to that? Because if we're decreasing the population size, we have to have some way of increasing it as well. So I also have this kind of DNA model. So in biology, the enzymes are built based on instructions contained within the DNA, and the rate that they're actually produced can be modulated by the presence of other enzymes. So those are the two factors here. Promotion increases the production rate of some number of target enzyme types, and then repression decreases that rate. And those are the operon binding relationships. Okay, so this is the kind of simplified model that I used for the first three, the ones where it's protein to protein one-on-one binding interactions. So here we have some protein population K, and it has an activating relationship to protein population S. So these two populations, K is capable of binding to S, and when it does bind, it causes this reaction to happen where inactive members of S into active members. So slowly over time, you can imagine all of the members of S would become active in this case. And so I base it, so I have a couple of my equations here. So P sub E of K, that represents the probability of encountering a member of population K. B ideal would be the ideal number of the population K that bound to the number of B ideal is the number of proteins in the population K which would be bound to the population S at any given moment. So you can see I'm just basically multiplying a couple of probabilities together here and then multiplying that by the population size of K. And then this is kind of the ideal case. There's a more complicated version for when you add in the complicating factor of having other populations competing and that sort of thing. But this is good enough for here. So this is the model that I used for the first three examples where it's one-on-one. And then and then I have the local binding model, which is used for modification of the gene expression, which changes the rate that the enzymes are being expressed. Yeah, we don't need to go too deep into that right now, but basically the ideal version of the equation is just removing the P sub E term because we're assuming that the enzymes are always in contact with one another. Okay. So when it came to actually building the model, I did it using a game engine actually, because that was what was available to me. I used the monogame framework, and that allowed me to because game engines have this like built-in notion of recurrent updating. I found that it was suited to building this model, and then so I wrote this program that could take a structure that I specified and then simulate what happens to that network over time. And then I designed some networks to illustrate what the actual computational abilities of the paint model are. So the major thing that I did was logical gates. So Boolean logic is the basis of modern digital electronics. So they're incredibly important to modern computers. They allow them to perform various calculations and vary behavior according to input, so on and so forth. So in Paint, I built what's known as a functionally complete set of Boolean operators, which basically means that it's theoretically powerful enough to do any arbitrary Boolean computation. So to actually look at what I'm talking about, so here I have the network structure for the NOT gate, which basically it takes in a signal, and if the input is 0, then the output should be 1. If the input is 1, then the output should be 0. So it's basically the opposite of whatever input you put in. And in this case, I'm encoding the input into the active fraction, so the proportion of the population A that is actually active. So I set that manually, and then see how the dynamics of there's this kind of competition that occurs between this population and this other population, and the result is essentially a NOT gate, and you can see the output over here. So whenever the input is 0, then the output is 1. Whenever the input is 1, then the output is 0 or close to 0. And I'll go into the issue of this kind of loss of signal here in a minute, because this doesn't quite go down to 0. So my next network to build was the OR gate, which is basically if A or B is on, then the output should be on. The output should be 1. If they're both 0, then the output should be 0. And it's a very similar kind of mechanism here. We have these populations that have computing relationships to the output. And then I have a graph of the output. And again, you can see there's some degree of signal loss here, as well as there's a small lag time. Actually, so this lag time represents the time that it takes to adjust to this change in the input. I actually set the parameters of the network in this case so that they would show that this lag time exists. But really, you could play with the parameters to make this lag time arbitrarily small. So that's just for illustrative purposes. On the other hand, on the signal loss, you can definitely play with that. You can never get that quite to 0, which is why I built this next network, the amplifier or conditional network, which solves this signal loss problem. So the way that works, that's the network structure, and we don't need to go into, this one's rather more complicated. But basically, it has this threshold value in green, which you can adjust. And then whenever the input, which is blue, whenever the input is below the threshold value, then it sends the output down to 0. Whenever it's above the threshold value, it sends it up to 1. So what it's doing is basically quantizing the input. And again, we have some lag time, but I teamed you can set that to be arbitrarily low depending on how you set the parameters of the network. And so with those three in conjunction, that's a functionally complete set. And you can do any kind of composition of Boolean operators. So you could build, for instance, an adding machine and so on. But essentially, a large set of computations are possible that way. And then finally, I have this network, the time delay network, and I don't have a structural diagram for this one. It's rather complicated, but anyway. But what this one does is that it has no signal for a while until it reaches some time, some adjustable time, after which it sends out a signal. So that's fairly simple. But with that, you can simulate systems like neurons, for example, which fire, and then they have a recovery period before they can send out a signal again. You could simulate systems like that using a time delay network. Okay, so how does this apply to the collective behavior? So one of the things that scientists in the field of collective behavior are calling for is this evolutionary theory of it. So why do systems like anthills and beehives evolve? And also, there are certain patterns that tend to evolve all throughout nature in these collective processes. For example, we looked at a negative feedback loop before in the lac operon. That's a very common network motif. And there's a number of other motifs which tend to emerge. And we want to understand why those particular systems evolve and in what situations they're optimal, in what situations are the best solution. So what I'm proposing is that if we apply sort of simulated evolution to the paint networks, where we simulate different environmental constraints, what evolves at the other end may reveal what network motifs are optimal for various constraints. So what I'm essentially saying is that paint can represent a generic example of a collective behavior system. So what if this works out, it would represent a rapid research method for studying how collective behavior evolves under different constraints, which is then, that kind of generates hypotheses, which then we can go actually study in nature. And that has the bonus, if we apply this sort of simulated evolution, these genetic algorithms to the paint networks, then we can also use that to automatically design and optimize them for application in other domains. So we can harness these networks to do not just like OR gates or NOT gates, but to do things, for instance, like robot control or stuff that traditional programming might be less good at. Okay, so yeah, like I was saying, we want to be able to automatically optimize these things. Part of the reason is that, see, I designed all the networks that I showed before by hand, but that's not really feasible for larger, more complicated networks. So there's this related field known as neuroevolution, which is using genetic algorithms to optimize artificial neural networks. And there's some similarities between that work and what I'm trying to do with paint. So I'm looking into adapting this algorithm called NEAT, which is a neuroevolution algorithm, to work with paint networks instead, which would then allow not only, like I said before, simulating different environments and then determining the effect on the evolution of collective behavior, but we can also compare the efficiency of paint to simple artificial neural networks. And we may be able to determine if systems like paint may be better suited for certain kinds of problems. Okay, so to kind of take a broader view as I wrap this up, I have this quote from the book I Have a Vortex, which talks about the evolution of the brain. He said, so the author says, "I propose that the mind evolved as a goal-oriented device that implements predictive or intentional interactions between a living organism and its environment. Such transactions, to be successful, require an instrument that generates an internal image of the external world that can then be compared with sensory transduced information from the external environment." So you can argue that this kind of applies to the cell. Like I was saying before with the signal transduction, the internal state of the cell, the internal image reflects the state of the external world. The internal state being the physical distribution of biomolecules, the state of DNA expression, and that kind of thing. And that varies in accordance with changes in the external environment. And that gives rise to behavior in the cell, which is aimed at survival. Yeah. Okay. So to kind of wrap things up now, what have I learned in the course of this research project and before? Well, I tried to sum it up in terms of these 6 points. First, passion and the importance of being interested in what you're doing. That because you really need a lot of like dedication if you want to do research in high school, and also mentors. They feed a lot into that. I know I've met a lot of people. Even if I've struggled with some, a lot of those mentors have been incredibly helpful in bringing information to my attention and keeping me motivated. And they've been really important on the journey that I've taken. My third point, independence. You have to do this stuff independently of the things that you're learning in class. You can't kind of sit back and wait for the information to come to you, because science is about creating your own ideas, and because you don't really know the answer beforehand, and you have to have the the initiative and the courage to kind of ask those questions and pursue them on your own. In much of the case, regardless of whether you find a mentor, you need to maintain a degree of independence. My fourth point would be peers and the importance of learning from them. They've been incredibly inspiring in the course of my high school research. Fifth point, understanding failure and not taking that too hard. It was too much of a setback, but using it as a learning experience to push forward. And then that pushing forward, persistence through both failure and success. You can't grow too complacent. Everyone else out there is working hard as well, and you don't want to be left behind. Okay, so if I had to give advice, I guess, to parents who are interested, or also to high schoolers, it would be to foster interest in your kids as much as you can, but also giving them freedom to explore their own ideas. I wasn't assigned my research topic or anything. It's always been, for me, searching for my own ideas through readings and through thinking about the information, whether it comes in class or out of class, and kind of trying to just play with ideas, whether or not they're going to, turn into some project in the end or not. Just giving the freedom to think, I think, is incredibly important. And then finally, encouraging participation in extracurricular programs, which provide both the education, the mentorship, and the peers, which I think is an incredibly important part. Okay, and that's pretty much it. So I don't know if everyone's muted right now, but if there are any questions.
Excuse me, I have a question.
Mm-hmm.
So did you do all these experiments at home with like your own computer and your software, or did you like go to a lab or something?
Yeah. So this project was all, just on my laptop. Basically, after the Tardigrade project, I realized it's much more convenient, for me at least, to do this kind of modeling just on my own computer. So I'm not very good at doing work in the lab. So.
Yeah, okay. So, and you got the models. Did you have to use any data from like papers, or when making the models? Sorry. Did you have any data beforehand that you used to help make your model?
Not really. I mean, I looked a little bit at enzyme kinetics and that kind of thing, but in the end, what I'm doing is very abstract and mathematical. So it's kind of a layer separate from the actual reality. I'm not interested in predicting like what an enzyme network might do really in an actual cell. I'm interested more in the behavior of the network as a whole. So.
Okay.
I have a question. You stressed that the importance of the mentor. How do you find the mentors?
Sorry, I didn't catch that question.
Yes. How do you identify and reach out to mentors?
That's a really good question. I personally got really lucky because my most important mentor to me was a teacher at my school. I just happened to take his class. But if that's not an open opportunity, then what I know a lot of other people have done is just you can reach out to professors at local universities. Often they may be willing to let you shadow in their lab, or like I've mentioned before, through different extracurricular programs that you can do on colleges. Then you can meet professors that way, and even if you don't end up working with them after the program, then that's a reference that you have.
So do you also mentioned that you have a setback early on. Where did you get the support? How did you overcome that setback?
You mean with the tardigrade project?
Yeah, your first project.
Yeah. So I spent pretty much all of my sophomore year on that, and it was pretty disappointing in the end that I couldn't get it started. I think a lot of it was encouragement from my mentor. I mean, I think I probably would have stopped earlier if it weren't for him. But I don't know. I think you just have to understand that to a certain extent, failure is to be expected in research, especially at this level. Yeah, I don't. It is tough, but I think it's a lesson that it's really important to learn for high school research.
I have a question.
Mm-hmm.
So, what kind of program languages and computing platforms did you use for your project?
Yeah. Okay. So my model actually wasn't very computationally intense. So I just use let me see if I can pull it back. I use the monogame framework, which is this cross-platform game framework, pretty much. And I wrote my program using C#.
C#?
Yeah.
Okay, thank you.
No problem.
Thank you very much for your very exciting presentation. So I have a question. As a high school student, you must be very busy. So how do you balance your research project with your coursework?
Yeah, that's a very good question. And sometimes it's tough. I think one big point is that the summers are a very important time to take advantage of. Like for instance, you get a lot of uninterrupted time in the summer, and that was why it was possible for me to do the if I can go back to it, the minimum project. That was a summer thing. So make use of your summers, and then also you really have to carve out the time. You have to treat it as a extracurricular commitment. For me, I like I mentioned, I spent a lot of my lunches working on it. And at times it's tough to be away from friends, but that carved out a consistent time every day that I could work on my project, as well as whenever I had free time, just working on my whiteboard at home or during vacations. What else? I mean, as long you do have to kind of keep good time management. I also ended up staying up late a lot of the time because I wasn't very good with that. But yeah, you just have to make good use of your free time and carve it out of your day when you can.
Thank you very much. Also, do you think this project helped you to for college application?
Oh yeah, definitely. The Regeneron results came out after I'd submitted most of my college, or all of my college applications, but then I ended up sending out an update to all my schools. And I wouldn't be surprised if it helped me get into a number of colleges.
Oh, thank you. Congrats.
No problem.
Congratulations.
Thank you.
I have a question. So what are you interested in to pursue, like in a college?
Yeah. I'm still thinking about that because I'm interested in computer science. I'm interested in biology and in math. I'm thinking I want to do a lot of research as an undergraduate, and I'm choosing between the computer science and the applied math majors right now.
Oh, okay. Sounds very good. Yes.
Thank you.
I just have one more. Just like to know your thought. So you basically use the cell as a lab to study the collective behavior. You also mentioned that on the neuron level, and obviously in the society on the individual level. How so you think they are all kind of follow certain similar rules or some similar logic?
Yeah. Like if you, so there's work being done looking at the common factors that influence how these collective groups evolve. So I definitely think that there's a lot of similarity, and they probably they probably operate under similar constraints. So you would expect to find common patterns throughout them, at every scale, really, whether it's cells or ants or I guess human society may be a little more complicated, or maybe maybe that's just me being a human. I think that maybe it's in the end we're just as we follow the same kinds of rules as ants, but
So if you would like to summarize using your research, what are the key patterns here? You mentioned the environment, some reward or something. Hard work.
Yeah. So that part of my research is still being planned, the actual testing of that. But some of the patterns that people have identified already, if I can walk back to that slide, for example, negative feedback loops are one example where an action eventually causes, through this chain, it causes this feedback which shuts off the action. And another example would be positive feedback, which is the opposite, where something just amplifies and gets bigger and bigger. It feeds back into itself. So those are two examples. For more complicated behavior, for example, there's I forget what species of ant, but they make this kind of looping patrol kind of thing, where you have one ring of ants moving in one direction and another moving in another direction, and that makes it resilient to breaking down in a certain way. Yeah, I would say it's kind of difficult to apply the analogy to human behavior, but I don't doubt that it can probably be done.
Okay, it's hard to generalize.
Yeah.
So what are the key findings from your research?
So I think the first thing would be that this paint model, which is based on population level interactions of enzymes, that this model is powerful enough to represent essentially any arbitrary computation that you want, and then that's my major result, is that you can express, you can build networks to do essentially whatever computation. The next major work would be to create an automatic automatic design system for that, so that you can have it generate networks essentially that solve whatever computational problem you want.
Any more questions? If not, let's thank Michael again, and wish you all the best in the coming years.
Thank you so much.
Great talk. Thank you, Michael.
All right.
Yeah.
Thank you for listening.
Thank you.
It's okay.
Thank you so much.
Bye.
All right.
Goodbye.
Bye.